Welcome back, everyone. So I hope the last video wasn't too much. And in order to keep things a bit light, this next one will be a little bit on the lighter side so that we still have time to digest what came up before. We're going to continue this idea of nomenclature, but actually what I want to do is build into this, this next assignment that I want you guys to do on designing a drug. And in order to understand that, we need to understand a little bit about the biological nature of organic chemistry, or also known as bioorganic chemistry. So what I want you to do is to study some different functional groups and recognize them based on what we talked about in the last lesson, and then apply them in a biological context. We're also going to be naming and drawing some compounds, the kind of standard stuff. So we're going to get into the properties a little bit of these functional groups that we've talked about, and mainly the ones that we're going to talk about, the types of functional groups that we're going to talk about are either acidic, basic, or hydrophobic and hydrophilic. All right, those are the four main ones that we're going to look at a lot. And I hope you've seen these already in other courses, mostly biology, of course, with the hydrophobic stuff. And also maybe in chemistry, you talked about acidic and basic molecules. In general, though, just to refresh you a little bit, an acidic molecule will donate a proton, whereas a basic molecule will receive a proton. All right, so here is the molecule of the day. As you notice, it's not just one, it's 20 different molecules of the day. Let's zoom in on one of these molecules. I want you to forget about what A is right now. A, we're going to refer to it as the side group, okay? How come I can't delete this? Okay, I think my PowerPoint's bugging, so let me just refresh it. There we go. Just going to delete that. Okay. Yeah, so we're going to call this a side chain. But I want you to notice that the rest of this, what do we have over here? This, other than this side chain, we have, okay, well, over here we have an acid group. And over here we have an amine group. Okay, so which one's a higher priority? Is it the carboxylic acid group or the amino group? Well, I hope you said a carboxylic acid group because it's the highest priority. And so what I have here is some kind of amino ... acid. All right, and that's what an amino acid is, and I'm hoping you've heard this term before. This term amino acid makes up a huge portion of biological molecules that make up proteins, and you have a lot of proteins in your body, and they're very essential to our life. And so the most basic building blocks for these proteins is one amino acid. And when you string a whole bunch of these amino acids next to each other, then that's what you get as a protein. All right, or a peptide if it's small, and a protein if it's big. Most of the time, when it comes to our body, though, it's a protein because it's a huge molecule. And so I want to get into that aspect of it today, into this whole idea of amino acids, because it's really important to how our body does things, and things like enzymes, for example, how are they able to catalyze reactions and things like that. So over here we have only one amino acid, but I want us to string together a few. In order to do that, I need to focus your attention on three atoms: this nitrogen, this carbon, and this carbon here, okay? And so I'm going to refer to this as the NCC, okay, for nitrogen, carbon, carbon. And an amino acid can string next to each other. Whoops. I don't know why my thing is doing that. Okay, my eraser is not working, so let me quit this and I'll be back in 2 seconds. Okay, hopefully it works now. So I want to focus my attention on N, C, and C. And what we're going to do is going to actually string a whole bunch of amino acids together. Again, not paying so much attention to the side chain for now. The side chain is actually the most important part of this, but I just want to put it aside for now. So we have this N C, and this C has, this is the middle C here, it's got a hydrogen, and it's got this side chain, which is, for now, I'm just going to call A for now. And then there's this C, this carboxylic acid group. Now, if I want to chain it together, what I have to do is I actually have to, instead of this OH here, I have to have an N again. And so then I repeat the whole cycle. So it's NCC, 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 NCC. And right now I have 1, 2, 3, 4 amino acids strung together. I want to fill in the rest of my atoms here. So let's say this side chain, we're going to call this the B side chain. And so this has to have a double bond O. This has to have a hydrogen. This is going to be the C side chain. This is the double bond O. And right now, whether it's up or down, it doesn't matter. Don't worry too much about that. So this is that. This is going to be the D side chain. And this is going to be the double bond O. And because this is the last one, then I can have my OH again. Okay. And so whenever we're talking about proteins, we're always talking about the N-terminal ends. So N-terminal end and the C-terminal end. And this is why it's called the N-terminal and C-terminal, because on one end is a nitrogen, on the other end is a carbon. And this whole thing is one huge long string. Now, real proteins aren't just 4 amino acids long. They're hundreds and maybe thousands of amino acids strung together. And then that just makes up one protein. And proteins interact with each other in clusters of 4 or 5 or 6 different ones. And they can be huge, and so they can get quite complicated. But how they come together depends entirely on this A, B, C, and D side chains, okay? Because otherwise, they're very generic, right? It's always NCC, NCC. If every protein in your entire body, and we have 20,000 or 30,000 different proteins in our body, if we have 30,000 different proteins and they all looked NCC, 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 then how would you determine which one's doing what? All right, or how would it even be able to do different things based on its specialization? Well, the reason for that is actually those hundreds and thousands of strings of amino acids strung together perform a special function based on the sequence of side chains that are on those amino acids. So let's take a look at those side chains. And first, before I start looking at them, I want to point out that we're still doing this NCC, NCC. NCC. And that side chain is always connected to the middle C. All right, I want you to be able to notice that, NCC. And I can do this. There's 20 different amino acids, 20 different common amino acids. There's a few other rare uncommon ones. We're not going to talk about those. But there's 20 common amino acids that make up all the proteins in our body. And each one has a different side chain. So for glycine, for example, the side chain is easy. It's just 2 hydrogen atoms. This side chain is a methyl group. This side chain is an alcohol. This side chain is a different kind of alcohol. And every single one of these will have a different side chain that has a different property. And those properties are what allow the protein to perform different functions. And so let's talk about those properties a little bit. Wow, my eraser is still not working. I don't know why. Let's just hope that when I restart my computer, it'll fix the problem. But for now, I don't need it. So let's take a look at some of these properties. We said that there could be an acidic or basic amino acids. So I'm going to go ahead and characterize all the acidic ones right now in orange. Orange is going to be acidic. And acidic means that it has a carboxylic acid group. Now you might tell me, well, Cash, all of the carboxylic acid, all the amino acids have carboxylic acid groups. You're right. They all do. So then there's no point in calling them acids then, is there? But what I'm talking about specifically is acid groups in their side chains. Okay? So in their side chains, let's take a look at which ones have a carboxylic acid group. So I have one over here. So I'm going to call this one acidic. And it's pretty easy. Look at that. In the name, it's written acidic. And this one here is also written acidic. So this one's an acidic side chain. Those are pretty much the most acidic ones, and then we have some of the more basic ones. Some of the basic ones will include those that have big amine groups. So for example, this one is pretty basic. It's got a lot of nitrogen. This one's got a lot of nitrogen. So basically all these atoms, nitrogens normally allow you to accept protons. So look at this, there's a bond missing there, just to make sure to add that. This one is basic. And for the most part, that's pretty much it. All right. Then there are the hydrophobic ones. So hydrophobic, I'll do that in green. Hydrophobic means that it's got no polar atoms. And polar atoms include things like oxygen and, well, in this course, we're going to stick with oxygen. So anything that's got oxygen in it, we're going to consider to be polar and therefore not hydrophobic. Hydrophobic would imply that it's got a lot of carbon and hydrogens only. So this one's got only hydrogens. So yes, this one is hydrophobic. This one's got carbons and hydrogens only, hydrophobic. This one's got an oxygen, that's hydrophilic. So let's already start a new color for hydrophilic. How about red for hydrophilic? Hydrophilic. So the polar ones are going to be hydrophilic. So this one is a hydrophilic amino acid. This one is hydrophilic. It's got an oxygen. This one's got kind of like something like an oxygen. It's a sulfur, which is similar to an oxygen. But we still classify these because the sulfur is not nearly as polar as the oxygen. We still categorize this under the hydrophobic category, even though it's maybe somewhere in between. Over here, it's only got carbons and hydrogens, so it's hydrophobic. Same here. Same here. Oop, same here. Sulfur, again, remember, it's kind of hydrophobic. Over here as well, it's got a ring, but it's still only carbons and hydrogens. Same here. Carbons and hydrogens are hydrophobic here. This one's got an oxygen, so this one's going to be hydrophilic. And it's hydrophilic, and the oxygen is hydrophilic because it can make H bonds, or it can make temporary polar bonds with water, and that's what we call it, hydrophilic. The next one is kind of hydrophobic, but at the same time, it's also a bit basic. So I'm going to also put in the blue one, and it's okay that it's blue, that it's both. It doesn't really matter. So these ones are polar for sure because they have oxygens in them. Over here, where it's polar again, so we're going to say hydrophilic. Hydrophilic. And you might say, oh, Cash, aren't those ones basic as well because they have amino groups? Well, these amino groups are not very basic because they're just adjacent to a double bonded oxygen. So that's enough for it to not become very acidic. Those nitrogens are not going to want any more hydrogens. They're pretty electron rich already. Over here, we're going to say that this is hydrophobic because it's got no oxygens. This one the same thing, and this one the same thing. Okay. However, I'll say that these three last ones are just mild cases of hydrophobic. They're not the most hydrophobic. Okay, now we have all of that. Now, why is this important? The reason that these are important is that because when you have a long string of amino acids in a straight line, it doesn't really mean much. A straight line molecule doesn't really exist. And the way the protein gains its function is by how this big long string of amino acid actually folds into itself. So imagine a big string and you crumple it up into some specific, very intentional configuration. And now that is a functional protein. And how that protein comes together is through electrostatic interactions and through hydrophobic and hydrophilic interactions. So things that are hydrophobic tend to go on the inside of proteins because the cell is made up mostly of water, so it wants to stay away from the water in the cell. So the hydrophobic regions tend to go inside the protein, and the hydrophilic part tends to be exposed to the environment. Then you have acidic and basic molecules, and as you know, acidic are going to be more negatively charged, and basic is more positively charged. So let's write that down here. So negative charge and this one is going to be positive charge. Whoops. And so they can interact with each other, right? Because there's an electrostatic interaction. Isn't that coming from electricity and magnetism, electrostatic interactions? So there's an electrostatic interaction between the acidic amino acids and the basic amino acids, because the acidic amino acids lose their proton and become negatively charged, and basic amino acids gain protons or gain hydrogens and therefore become positively charged. And that pretty much summarizes what these amino acids are, how they're different, right? So remember, all these side chains are different, or all 20 of them are different, and it's the combination of them that produces the specific functionality of a protein. So that being said, I'm going to stop there because that's the knowledge that you need in order to understand the assignment that we're going to do in class next time we see each other. That's it for now. I hope that was clear. If you have any questions, please let me know, and see you in class.